I have a lot to cover today, and I'm going to try to be as fast as possible. I assume that many of you know some of the background that is listed in these slides, and so I'm going to go relatively quickly on the first piece, but I'm going to talk on progression of aging, biological aging clocks, first and second gen, the Stanford 1000 Immunomes Project that I've been leading for the past 17 years, functional aging clocks, system-specific clocks, and last, space fuel analysis of aging trajectories. So the first concept I'd like to stress is that aging is hierarchical. We see initially at the age of 25, 30, 35, changes at the molecular and cellular level, and these are the so-called hallmarks of aging that all of you know about. That follows dysfunction in different tissues and organs with fibrosis and arterial stiffening and stuff like that, that we can put in one bucket, which is basically phenotypic aging. And that leads to dysfunctional systemic aging with lower intrinsic capacities, frailty, and stuff like disability. And so we've been very interested in developing ways to measure every single step along the way with different technologies. What I'm showing you in this slide is a collaboration I have with Joel Kramer at UCSF. We collected data from 1,300 people, and this is essentially to illustrate that the gray matter volume decreases with age. That's phenotypic aging, but you can also see functional decline, things like cognitive impairment, mild cognitive impairment, or even early onset dementia, which is already a disease. So I want to convince you that there is this progression of aging from biological to phenotypic to functional aging. I'm going to start with biological aging, and I don't have to explain too much because yesterday there was really good talks from the team, from Steve and Vadim and Nora, looking at epigenetic clocks for the most part. Suffice to say that these are changes at the level of the DNA with methylation, CpG islands, and these methylation can be demethylated and methylated, and so there's changes, and those are so strongly correlated with aging that you can create these chronological aging predictors. These are the first-gen clocks. And then you use that variance to try to estimate disease risk and to evaluate responses to different age-related interventions and longevity interventions. I apologize that this didn't come very clearly, but this is work from Wang here in China et al., showing, this is basically a simple review, to show that first-generation clocks started here in 2012 with the Hammond clock and then Horvath clock, and they evolved over the years to second-generation clocks that are predictors of mortality or morbidity or even functional aging, as I'm going to show you in a moment. I'd like to highlight that inflammation is one of the key elements in aging as it relates to different hallmarks. As is shown here, we published this in Nature Medicine and Molecular Metabolism. There are very tight connections between all of the hallmarks of aging and inflammation. And so as part of the 1000 Immunomes Project, we focused on developing a tool that can measure inflammation in the body that is associated with aging. Very different from acute inflammation. When you cut your finger, you have warm and red and swollen signs. There are clear markers for acute inflammation, interleukin-6, CRP, but for age-related inflammation, there's no such thing. And so we came up with a deep learning method to predict chronological age with the spirit to develop a novel first-generation clock. And when you take the residual, we can correlate that with multimorbidity. We predict frailty 7 years before it kicks off. We can correlate that with immune function. These are a large number of different stimulations that we did in peripheral blood mononuclear cells from the 1,000 individuals in the study. We correlated that with ventricular remodeling, exceptional longevity, and we can predict mortality in independent data sets. But this test and many other tests that are available to estimate biological age are invasive. And Jackie did a great job at showing yesterday her 3D technology. And we published this with Vadim and other collaborators, illustrating that skin health and appearance are tightly linked to other hallmarks of aging in the body, in the blood. And there's bidirectional communication between the skin and the blood. And so what we did, we leveraged data from one of my clinical trials that we run in one of my companies with over 600 individuals. And we used frontal 2D images, basically a selfie that you can use with your phone. We did essentially a deep neural network, CNN. We did PCA. We concatenated this after cropping and doing all the fancy AI with the metadata. And last, we're trying to predict these proteins and inflammatory age from the blood. This is work that Minia in my lab is doing, and this is unpublished data. But you can see that we can more or less predict inflammatory age from the photos, and we're basically having now a tool, ultra low cost tool that could indicate with 65-ish% accuracy if you have accelerated aging in the blood or not. And when I was traveling here, I gave it a try to the Healthy Selfie that we call, and unfortunately my photo iAge is higher than average. I didn't feel very well about that. But this is something that we're now implementing, and we're soon going to make it available for everybody. Not only we could predict inflammatory age, we can also predict the levels of proteins. That's shown here, different models with and without gender. And we used 95,000 images from different ethnicities, Black, East Asian, Indian, and other ethnicities, Asian, and so forth. And these are the distributions that follows a normal distribution. The iAge predicted from the photo correlates well with chronological age, and the residuals are shown here. So moving on to a different area, we explored soluble factors that can impact brain function. And after a lot of work with Joel Kramer, we came up with a model that can predict, again, first-generation clocks, chronological age, and after adjusting for it, we can run a canonical correlation analysis and decouple the effect of age, sex, and the cyclo, which is the cytokine clock. And what you see is that age is most correlated with sensory motor, so sensing environment, motor skills, and so on. Sex is mostly correlated with executive function. Women actually do better. So I decided my next CEO will be a female. And cyclo correlates mostly with emotions, motivation, memory that are all part of the dorsal anterior and limbic systems that is largely affected in Alzheimer's disease. So moving on to the functional aging clocks that I promised to show you, if I had a lot of time, I would ask the audience who believes that aging should be considered a disease. Probably this audience will say, some of them agree, probably 5 to 10% in the audience will say yes, the majority will say no. And there's political reasons, there's social implications, and so I don't think it should be considered a disease, but in 2022, we had the first code, the MG2A, which considers age-related decline in intrinsic capacity as a disease in the International Classification of Diseases, ICD-11. What is intrinsic capacity? Is the basis for maintaining function. And so what you see here is you can actually measure those things, psychological, vitality, cognition, locomotion, and sensory. We run a clinical trial. This is a 10-year longitudinal study of intrinsic capacity in collaboration with Professor Bruno Vella. And we got a lot of different measurements on these individuals with cognitive status, performance, physical, and then grip strength and other things. And we also had biological measurements like DNA methylation in the same individuals. And what you see here is that different domains of intrinsic capacity decline with age. And you can see the model performance in predicting overall intrinsic capacity using this DNA methylation profile. So with 92 CPGs, we're able to predict intrinsic capacity, and this is the age correlation. It's very interesting to see that almost none of the CPGs overlap with canonical clocks, like the Hannum and Horvath and PhenoAge. So we're discovering new biology here. And what is this biology telling us? The biology is telling us it correlates, or it's driven by immune function. So the higher the intrinsic capacity, the better the immune function. The lower the inflammatory age, the iAge I already mentioned, C-reactive protein and IL-6. And we could confirm that with LLM models that now we're using to associate gene sets with different hallmarks of aging. In people with high intrinsic capacity, they live 5 years longer. So this is predicting mortality effectively. And what you see in this slide is that certain lifestyle decisions and nutritional features can impact intrinsic capacity in different ways. Fish and omega-3 indicates better intrinsic capacity. I don't have to convince you to eat your fish because I know it's a very fish-rich diet that we have here. I'm going to move on to organ-specific clocks. And this does not differ too much from the work that Vadim was showing yesterday. And why are we doing this? Well, there's a very simple reason. Overall, systemic epigenetic age is a very poor, low-resolution indicator of health of each organ. So see, for example, in this case, we see biological age acceleration is 0. The net is 0, but you see that the heart has an acceleration, so this is the Achilles heel. We are decelerated with the lung and the brain, and then if you sum up, you'll have a result that tells you this person is doing well. However, there are certain areas for improvement. So we went ahead and leveraged the data from the UK Biobank and the Health and Retirement cohort, and this is work that we're doing with Eileen Crimmins at USC in California, and tweaked some of the previously reported papers to come up with a predictor of organ aging or system-specific mortality predictor using simple lab biochemistries. So nothing fancy, just the regular lab biochemistries. We can create these different predictors of mortality that then we transform to an age or biological age of each organ. And you can see this is the accuracy, so the C index, the concordance index in predicting mortality is overall very good. And you can see which features contribute the most to this organ age indicator. We have 33 different labs, and unfortunately this didn't come out very clearly, but I love this one here. It's one of the most, I would say, inspiring figures, and you can't see it, so I'm going to explain. Here you see the correlation with age, and here you see the C index. There's no correlation, which means that seeking extra accurate clocks does not mean they don't necessarily correlate. The accuracy of the clock does not correlate with the quality of the prediction of diseases or mortality. So don't search for perfection when you build a clock. And then similarly to what Vadim was showing yesterday, if you have acceleration of the circulatory system, you're going to have a high chance of developing angina, congestive heart failure, and heart attack. If you have acceleration of the mental system, high chances of developing dementia, and so on and so forth. Because we had access to proteomics data, to metabolomics data, and transcriptomics and epigenetics, we can build proxies. We can build proxies for each one of these organ aging indicators or mortality risk predictors. So what you see here is proteomics, metabolomics, transcriptomics, and epigenetics. Overall, we see around 0.75 C index, so it's a little less accurate than the original model, but if you have epigenetics in hand, you can calculate this as well. And this is validation, and this is looking at different lifestyle choices. And it's very surprising to see that red wine has a negative impact, meaning that it drives deceleration or is correlated with lower age acceleration, but things that we know very well are terribly bad for you. We validated it, white bread, burger, margarine, and this is probably the white bread that we see in the United States, full of sugar and other additives. We can also see that exercise is very beneficial. Overall, the metabolic system decreases as a function of how many times we do exercise. There is a plateau, and there are other systems that benefit as well. And you can see that smoking is logically not a good thing. Smokers have a high chance of having age acceleration for the respiratory system, but there's other systems that are affected as well. So smoking leads to higher age acceleration for all of the different systems, but the respiratory system gets affected the most. So I'd like to move on to a project that I started around 4 years ago with NASA. And they reached out to me because they seek for a collaboration with Susie Sanello, who said, David, your research in the immunome and aging is so inspiring, and maybe we can do a collaboration to help our astronauts do better. So what you see here is a cross-sectional data. This person seems to be okay. This person seems to have an acceleration of certain clock. It doesn't matter which one. Now look at the 1,000 Immunomes longitudinal study with 5+ visits. This person actually developed heart attack, started accelerating very fast, and this person who one would believe has a high risk actually did quite well. So could we predict those trajectories? And what we did was to use the learnings from these 5 papers that we published all at once in the Nature packet last year on looking at age acceleration, on space travel and microgravity. We envision systems to simulate microgravity on Earth in the lab. And we're using here different organoids and PBMCs. From the organoids, we subject these to microgravity simulation. And we do a whole slew of different functional features, and we also measure rates of aging and hallmarks of aging. And you see that neural organoids have an acceleration of aging, cardiac organoids as well. They get affected in different ways, in different hallmarks of aging. And we also could assess different functions of these organoids. They're really, really fun to work with. Brain organoids can actually be taught. They have fire potential. They're very cute. Heart organoids are mini hearts that are beating. And we can simulate or emulate Parkinson's disease, cardiomyopathy, and immune aging. We vaccinate tonsil-derived organoids, and we have a really good model there. With the PBMCs, we use it for wellness and health and wellness space. So we developed a product called bioIage, and this bioIage is an overall pace of aging. Imagine you have baseline and microgravity. So now you have 2 time points. You can draw a curve. You can draw a pace of aging. and have that for every hallmark of aging. And we're giving individuals another blood draw with a Tasso device as a follow-up in 3 months to make sure that they fall below this curve and they're doing better. So in summary, these are some of the clocks I mentioned, first gen and second gen. And this is a very fresh slide that I want to maybe stop for a moment here. We have predictive horizon index. This is something we came up with. It was essentially a population-based assessment on how good the clock is at predicting long-term health. And you have estimated years of life. This is individual level. For a person 50 years old, you have the estimated 25 years of life. And so you see that different labs can predict relatively well that individual trajectory, but with our clocks and GrimAge as well, this is disease age and GrimAge, we're doing much, much better at predicting long-term health. This is 9 and 10 years out. So very, very relevant for actuarial sciences. So that's the summary. It's basically the same as my outline. And I want to thank my team. We do a lot of things other than science. We go hiking and surfing and party. Thank you so much for your attention.
Thanks for the speech. 请观众朋友们踊跃提问。
Very impressive work. Congratulations. This finding about red wine is amazing. So how much red wine should one drink per week to have a benefit?
Okay, we can chat later, but my take is we are looking at feature by feature, just like every single epidemiological study has been done in the past, and I think that's probably not the best way to go. We need to understand clusters of behaviors because wine comes with other things, comes with a connection, comes with talking to others, comes with de-stressing. Maybe there's no single element in wine that is bringing this down. And so I'd like to just take this with a grain of salt. But what we see is that overall, there's like the typical recommendation with 1 or 2 glasses of wine that causes this decrease that is significant for all of the systems, as you could see. It's very significant. And this is in thousands of people, dozens of thousands. Thank you, Steve, for the question.
Thank you.
How do you make sure the accuracy of the facial imaging prediction for the whole body inflammation aging? And do you also use other parameters? You know, nowadays many people love to make surgeries or laser treatment, other things on our face. So I'm so curious about it.
Yeah. So the accuracy is measured by estimating acceleration or deceleration. This is an area under the curve, very simple classification, and this is cross-validated, okay? So that's the 66% accuracy that I showed you before. Does that depend full on the images? No, there's a small percentage that can be explained by the hip measure on the in centimeters and the waist. So those contribute about 2-3%. Which we didn't have in the public images. We only have the photos. So we have a lower percent accuracy, but we could demonstrate that the distribution of iAges predicted from different ethnicities remains pretty stable, pretty constant. So there's, I hope that answers the question. Yes? Great.
Maybe I missed something. How did you measure intrinsic capacity?
So intrinsic capacity we measured in the clinic. And there's the very typical battery of tests for cognition with MMSE and the MoCA test. We have for frailty or for vitality, we have grip strength, all these different things. And for psychological, there's a questionnaire. For nutrition, there's also a questionnaire. And then hearing and vision.
Sorry, another question is regarding your lab test. From the lab test, you inferred all this organ-specific clocks, right? So you assign the different parameters in the lab test to different organs?
No, we don't supervise this. It's completely unsupervised. We take all of the labs and we're predicting mortality. It's a mortality risk estimator. Then we convert that to biological aging by scaling against chronological age. But in essence, this is a mortality risk estimator based on the labs. And it's whatever the model's picking up.
Very interesting, David. I wonder what is the contribution of various modalities in predicting disease age? Like, is proteomics still the major one, or like how does metabolomics fare, or other kind of measurements?
We didn't try combining these modalities with labs. What we tried was to take all of these omics and predict the disease age score for all of the organs. Turns out that the metabolomics is the weakest predictor. Then epigenetics and transcriptomics are very similar, and proteomics actually beat all of them. But again, the model was trained on labs. So these are proxies for the scores.